You have a worksheet that looks like this, and I have taped little papers on top of mine because I'm going to use different information than what's on here. I'm not going to give you the answers, but I also don't have a printer, so I can't print it out again. So that's the explanation for this. So the first thing is to find the opposite side of the DNA. The template strand is provided for you. So let's say that our end goal is lactase. Lactase is a protein, and it's the enzyme that breaks down lactose. And if you don't have a functioning lactase protein, your body can't break down lactose. You're lactose intolerant. So first of all, let's find, crap, oh, here it is, okay. No, blue, okay, well, blue will work. Let's find the opposite of this strand. We're making DNA, remember. So we have 3' here, which automatically means that this side is going to be 5' because DNA is anti-parallel. Opposite T, we have A. Opposite A, we have T. Opposite C is G. Opposite G is C. So let's say that this is the information we want. This is the information that's going to tell us the order of amino acids in the protein lactase. So in other words, the information on this side of the DNA makes sense, so to speak, for the lactase protein. So this side of the DNA is called the sense, or the coding strand. And you can see that on your worksheet. One strand has been designated the coding strand. It's the opposite of the template strand. The other side of the DNA is the template strand, and that's what we're going to use as a template to get the information. The template strand is sometimes called the antisense strand. All right, so let's make some mRNA. Remember, the RNA polymerase is involved in the first step of protein synthesis. That's transcription. RNA polymerase opens up the DNA, there we go, and finds RNA nucleotides and adds them on based on the order of the template strand. So let's do that again. 3', we've got 5', and this time we're making mRNA. All right, this is the second step of your worksheet. Opposite T is A, opposite A is T. Oh, just kidding, U, because we're in RNA. C is G, G is C. So we're finished with transcription. This is your mRNA. The RNA polymerase read the template strand, but wrote the information found on the coding strand, the sense strand, because this is the information we need to make lactase. This is not the information, this is the information. Look how it matches up almost exactly. A to A, U, because RNA, T, G, G, C, C, A, A. This is the information we're getting, and we're getting it by reading the template strand. End transcription. Leave the nucleus. DNA goes back together. Goodbye. Now we are in the cytoplasm. Turn the page. The ribosome is the enzyme, the protein involved in translation. And the first step of translation is to find, so remember, mRNA is written in groups of 3 letters. Each one is called a codon. So here's one codon, AUG. The next codon is CAU. The next codon is UGG, and so on and so forth. Finds tRNAs. Remember, they're sort of like little taxis for the amino acids. The tRNA has on one end an anticodon, and on the other end it has an amino acid. So we're going to find, as the ribosome, the anticodon that matches with the codon, UAC. So this is the right one. And that's how the cell knows that methionine is the first amino acid. Methionine will also always be on a tRNA that has this anticodon, UAC. All right, now let's do the next one. Okay, AUG. CAU is the codon. So we're looking for, oh, it's right on top. What do you know? One with the anticodon GUA. This tRNA is holding histidine. A tRNA with the anticodon GUA will always hold histidine. All right, next one, where were we? UGG is the codon. What's the anticodon? Oh, there we go. ACC, that's tryptophan. So we've got methionine first, we've got histidine, and we've got tryptophan. All right, you guys out of the way. These guys are going to go back and get more amino acids put on them. This one will always have histidine. This one will always have methionine. Pause. Okay, so how does this all relate to your worksheet? Well, page 3, translation. First thing you do is you take the mRNA codons from page 1. So you flip back to page 1, right here, and you take this and you write it here, the mRNA codons. The anticodons are just going to be, you can just write down the exact opposite of what was on here. So the opposite of A is going to be U, opposite of U is A, opposite of G is C.
Go.
All right, so here I've written down my tRNA anticodons, and because I apparently made my example a little too long, I had to draw myself another tRNA on the end. So yay me. So that's what you're going to do. And I want to caution you against a common mistake, which is instead of writing U, the opposite of A, people will write A. Or whenever you see U, you would write U. Don't do that. U goes opposite of A, always. All right, great. So now we have our tRNA codons. But if you don't have this elaborate little setup, how are you going to know what amino acids to put here? Aha! That's where this comes in, the genetic code. This contains all the possible combinations of C's, G's, A's, and U's in groups of 3. So all the possible codons, there's 64 codons. And you might be saying to yourself, well, there's 64 codons, but there's only 20 amino acids. Well, that's okay, because multiple codons can be for the same amino acid. So for example, over here for leucine, we have CUU and CUC. Oh, and then there's also one up here, UUA. Okay. Let's go back over to part 3 translation. mRNA codons. AUG is the first codon. So we flip over. The first base is A, so we know it's going to be in this row. The second base is U, so we know it's going to be in this column. And there's AUG, methionine. Met. So let's write met. right there. Let's do it again. The next codon is CAU. All right. C is the first base, so we're going to be in this row. A is the second base. We're going to be in this column. Somewhere in this box, CAU, that's histidine. So we go over here and rewrite HIS, the abbreviation. Part number 4, mutating your gene. Below, rewrite the DNA message from the previous page except change one base. Cool. So we rewrite the DNA right there. We're going to rewrite this here. So I've rewritten the information that I would have written here on page 1. Just copied it exactly. And now I'm going to change one base. That will be a mutation. I think I'm going to change this one. I'll change this T to C. Yeah, that sounds good. And now I have to change the opposite one. Opposite of C is going to be G, so this is going to have to change to a G. Now, I'm going to put a little asterisk over where I made my mutation so that I can remember which one I changed. Turn the page. Transcription. What is in here? Okay. Transcription. Below, write in the mRNA sequence of the bases that would complementary base pair to the new template strand of mutant DNA. So we go back over here, we look at our template strand, and find the mRNA that would code for that. In this case, it's this mRNA, because I'm using that example. And then you write down the opposite of the template strand.